Hey guys, how's it going? Can you guys go to the classroom and take a look at the PowerPoint and the notes and all that stuff so that we can get going as soon as I'm done with attendance? Looking at chapter 13, meiosis. Hey, do we have... Sanjana, are you in? Okay, Sanjana Preet is absent. Everybody else looks like they're here. I'm sorry, I turned the waiting room off. Like, oh, Ryan Nowak, are you here? Ryan? I got to turn the waiting room back on. That's a lot easier to take attendance. This takes too long. Okay, sorry about that. So get your stuff ready. I'm going to do attendance real quick, and then I will be back. All right, your multiple choice tests are in. Actually, I don't know if they're in classroom. Let me double check that. So the multiple choice part should be done. I think I put it in Infinite Campus. Your free response, I've not had a chance to look at yet because I had some unexpected complications this morning. Let's see what we got here. Oh, I did import them. So why don't they show up here? Hold on a second. Oh yeah, they do show up. Okay, we're good, we're good, we're good. So you guys know what you got. I made the cutoff 11. So if you got less than 11, that means you did not pass. That means you should, or even if you got 11, I mean, you could still come in for TSS. If you could stop by for a few minutes today, I'm not going to make you like redo stuff today, but I'll tell you at least what you know, we should make a plan about what to do for remediation. All right, we got one more coming in. Oh, it's Ryan. Hi, Ryan. We shouldn't have to be here today. Why? I'm here. Let me see. Ryan is here. Work, present, save. Okay. Well, most people did pass, and some people got really, really good scores. So I think overall they're passable. You and I can talk individually about maybe what needs to be done and some strategies and things like that. Okay. Oh my God. How's everyone? Hey, Martina. Shouldn't have to be sleeping. Didn't you sleep last time and early today because you're not like seeing us? If it was based on how much I like you guys or anything like that, we would never even have class. Like if that was the criterion I was using of how much I like you, we would never actually meet. Anyway, so we can't use that one. So you can try to be more likable, and I'll make my tests more passable, I guess, or something. I unfortunately cannot let you leave early, but I am indeed planning Kahoot. I am planning Kahoot. So that is in the works this weekend. I made one, yes, because you guys said you wanted that. You guys told me you wanted that last week, so I figured let's try it out. I'm the most likable. I don't know about that. All right, let's talk about meiosis. This should be overall pretty easy. A lot of it will be reviews. Some of it will be new, not too bad. But I will say this. I'll warn you with this that meiosis can be confusing. Even the what we call easy parts can be confusing because it's easy to mix things up and it's easy to get away from that from the truth of it all. So obviously there's an obvious comparison to mitosis, and there's a lot of similarities, and that's nice. But the problem is sometimes that is a trap because we think it's just as easy as mitosis, and it's a little bit more tricky. So we do have to worry about things like sister chromatids, and we have to worry about things like homologous chromosomes and stuff like that, and what's the difference. And we have to be really sure that we know what the difference is and why all of this is happening. Why do we do mitosis? What's the point of it? And then why is meiosis necessary? Why do we even need it? And maybe we could start with that. We could start with that discussion. All right. Let us, I'm having trouble with my setup here. I'm sorry. Oh, that's not what I meant. That's not what I meant to do. What am I doing here? Oh yeah, here we go. PowerPoint. There we go. We're cooking. We are cooking with gas. Let us talk about the purpose of meiosis. So if we just revisit mitosis for just a second, we know that it had basically three main functions. Well, two main ones with us, but if you look overall with all living things, three main functions. You guys, real quick, throw them out, like just yell at me. What were the functions for mitosis? Just shout them out so I can keep moving here. Because keep in mind, kids, the faster you do this, the more active you are, the more time we'll have for the Kahoot.
Growth.
Are you saying
So growth.
Repairing of tissues.
Growth and development was one. That's obviously important when you're growing from a little tiny baby to an adult stage. Okay, what else?
Repairing.
Repair. So when something goes wrong and you break something, then you need mitosis to fix it. Like if you have a cut and it's got to heal or something like that, and it grows back. And then the last one was just reproduction, and that doesn't apply to us. That's like for amoebas and single-celled creatures. So, but those are the three main functions. Well, meiosis now is also a cell division, but it's a weird kind of cell division where strange things happen. Let's take a quick look at how most, and I can say most because it's true, living organisms reproduce. The answer is sexual. So we have a sperm, we got an egg, and if we're looking at a human and you got if you got 46 chromosomes in the sperm and you got 46 in the egg, which is how many chromosomes we have in our cells, by the way, the number of our chromosome number of diploid cells is 46, we would end up with an offspring that would have 92 chromosomes, and that would be a big problem. you know, our babies do not have 92 chromosomes, and their babies then would have, hold on a second, their babies would have, okay, go take care of it, but like try to stay in earshot if you can. Their babies would then double again. Like, let's assume you could make a human with 92 chromosomes in the cells. Now they produce sperm, they produce eggs, they produce gametes. So now you got 92 plus 92. Now it gets real ugly. You got 184. If I did the math right, I don't even know. I think that's right. You got 184 chromosomes. All of a sudden, it would keep doubling, and it would get unmanageable in terms of the amount of DNA that's in these cells. The nucleus wouldn't be able to contain it, and we know that abnormal chromosome number can actually cause serious problems with the organism. So we know about chromosomal abnormalities causing things like Down syndrome, like Cri-du-Chat, and different, a host of different abnormalities that cause serious problems with the person's well-being just because the chromosome number is wrong. So what does meiosis do to fix that? We go from 46 to, go ahead and yell it out.
23.
We go from 46 to 23. That's the solution. So it's a reduction division. It's actually two divisions. We only duplicate the DNA once. So technically we're technically going to 92 chromatids, because each of those, X chromosomes has 2 chromatids on it. They're 2 sister chromatids. We're going to divide once. We're back down to 46. We'll divide again. We're down to 23. And that's basically what meiosis is all about. It makes gametes, eggs and sperm. There are 23 chromosomes. They're haploid instead of diploid. They have half the normal number of chromosomes. That is the punchline in the big picture of meiosis. So understanding that is crucial. Now let's talk about all the other stuff. Let's talk about all the minutia, shall we? So in order to understand it fully, we need to understand the nature of a chromosome. So we have to understand that when we draw an X like this for the chromosome, that's actually 2 chromatids. And the only time our DNA looks like that is right before mitosis. This is not normal for chromosomes to look like these X's. That's not normal. That's actually duplicated already, and we have these sister chromatids that are identical, photocopied, replicated during the S stage of the cell cycle. Okay? And what you need to understand is that for each of the chromosomes 1 through 23, you have 2 versions. You have 2 of them, one from mom, one from dad. Because we're sexually reproducing organisms, we have shared combined DNA. We have some from mom, half of them from mom and half of them from dad. So that's what makes us diploid. Okay, so what we're looking at here is this picture right here is actually, it's a little misleading. This is actually 2 sister chromatids attached together. So our X is right here. Okay, so here's our little X or whatever. And this is another 2. So these 2 are really homologous chromosomes. Homo meaning homologous chromosomes, homo meaning same. So they're not exactly the same, but the one from mom and the one from dad are overall going to be very, very, very similar. They have all the same genes and all of that stuff, but slightly different versions, slightly different alleles of some of those genes in some cases. Like mom might have the gene for blue eyes, dad might have the gene for brown eyes. It's still the eye color gene though. That's actually a lie because there's like 11 eye color genes, but if we were going to simplify it like that, you get the idea. All right, so no same, okay. All right, so this would be homologous chromosomes, these 2. And then this right here, that and that is actually, that would be the sister chromatids. And if you do a karyotype, so here we have a karyotype. A karyotype is just taking a cell that's undergoing mitosis from an organism and squishing it and then spreading out the chromosomes on a slide and looking at them with a microscope. You can actually organize those chromosomes by size and by number. And the biggest ones are like in the beginning. So 1 is the biggest, 2, 3, all the way down to the smallest. The only exception is the X chromosome, which is the sex chromosome. That's big again. But the Y chromosome is really, really small. That's this one. Oh, I'm drawing black on black. That's dumb. Why am I doing that? Okay, so here, the Y chromosome is really tiny. That's what makes a male. And then the X chromosome is pretty big. And that's obviously in males and females, but XY is male and then XX is female. Well, that's all stuff you know already. I mean, that's stuff you learned about before. All right, so you need to understand the nature here of the DNA and the chromosomes. For example, when the chromosomes look like this shape, they're coiled up. The DNA is coiled up. It's not functional at this point. It's getting ready for mitosis. It's not functional. All right, cool. So this just kind of shows how there's two sets of each chromosome, that there's these homologous pairs, you know. So this would be like number 1. You know, here's like number 2. I'm sorry, here's number 1. Sorry, this is number 2. This is number 3. You know, and this organism, they're only showing 3 pairs. We'd have 23 pairs like that in a human cell. All right. So 1 and 1 would be homologous. 2 and 2 would be homologous. 3 and 3 would be homologous. Like I usually I'm kind of a traditional sexist kind of person. So I say like the red ones are from mom and the blue ones are from dad. Obviously that doesn't fly today, you know, like any person can have any color or whatever. But I mean, that's just what they did too. They said the maternal ones are red. They're sexist just as much as I am. And the paternal ones are blue. That's a good way to keep track of it, but it doesn't matter what color you use or whatever, as long as you understand that the notion of where our DNA came from and, you know, why are the chromosomes arranged like this. When you divide these cells, you have to divvy up the chromosomes very, very carefully, that each of the daughter cells gets the correct amount of DNA and gets all of the genes, so that one of each chromosome at the end when it comes to meiosis. So that's what this meiosis business is all about, is figuring out how are we going to do this? How are we going to arrange these so that this actually happens, so we don't mess up the gametes, which would then mess up the offspring and create something that's not suitable for survival. So this just shows the mammal life cycle. We got egg sperm, which is the haploid. I really should switch back to a real color here. Yes, Sim, there are naked people on here. Got to get over it. Just chill out. So haploid version is 1 set of chromosomes. It's just 23 in our case. Yes, exactly. OMG, chill. You got it. And then once the organism, you know, once the baby forms or once the zygote forms, egg and sperm combine, it's then diploid, and our main form or our main existence is in the diploid stage. And that sounds weird, like, well, obviously eggs and sperm aren't, you know, people, and they're not. They're just cells. They're sex cells. The people are the diploid stage. But in other organisms, there's haploid organisms where the actual organism is haploid. It's super weird. So let's take a look at that. I assume you know what ovaries and testes are, so I hope I don't have to go into that, but sorry. So here's the mammal, animal life cycle, where the haploid stage, so the gametes are a very, very short-lived stage. Okay, we just have the egg and the sperm for a very temporary amount of time, and then they come together and form the organism, and the diploid stage is then the dominant stage that's throughout the whole life of the organism. It's what forms the organism. All right, so diploid is long-term. And the haploid stage is just unicellular. It's just 1 cell. And this is multicellular. Obviously, you know, we're multicellular. We have a lot of cells. So that's normal to us. Like, this is like, well, duh, obviously, I already know this. Why are you telling us this? Well, the reason I'm telling you this is because it's not obvious for every organism. So there's some weird organism, like some plants and algae. They actually have two sort of stages that are pretty long-term. So that the haploid stage of a plant could be a fairly long-term stage where it actually mitosis happens with the haploid cell and a multicellular organism forms that's haploid. That's weird. It's like as if, imagine if sperm like grew and formed this like multicellular organism that was running around and then looking for, I can't even, I'm sorry. I shouldn't have told you this right before break. Now you're gonna have nightmares. Yeah, but anyway, but that's kind of what this is. It's like these haploid cells growing and forming a multicellular creature. It's super bizarre. All right, and then the diploid stage is another part of the life cycle, and that's, you know, another long-term stage that sometimes they live together, where the haploid stage, or actually it's the diploid stage, lives on top of the haploid part of it. The diploid part grows out of it. Very bizarre. Well, plants are weird. You probably already knew that. And then the most bizarre of them all is what fungi and then some other protista do this. Their haploid stage is the dominant stage. So if you look at a mushroom, you know, if you're picking a mushroom in the woods or whatever, the mushroom part, that's called the basidiocarp, that's actually haploid. Like that thing you pick up that's up above the ground there, that's actually a multicellular haploid organism. And the diploid stage is very temporary. So the diploid stage is real short term in these guys. It's very, very odd. None of these things I just told you I want you to necessarily memorize. I don't need you to memorize these life cycles or anything like that. I just want you to be aware that things are weird. Nature is weird. And what we take for granted as like what's normal for us is not necessarily normal for fungi and for plants. They do bizarre stuff. Mushrooms are yummy. Mushrooms are disgusting. This is very informative. Thank you. I knew you did drug. I knew you did drug. Yeah. Anyway. That kind of sounds similar to what my son asked me. I must have said something weird or whatever. And my son asked me, are you drugs? I'm like, I don't know, whatever. Put them in my fettuccine. The drugs? You drug your own? That's horrible. Do I need to call like the social services or something? Do I need to call the police? Oh, the mushroom. Okay, I got you. Yeah. That wasn't super obvious. All right. So in meiosis, the super ultra simplified version of meiosis is here. Odd things happen right in prophase. So we have two divisions. We have, you know, division 1 and the division 2. So we have a name for each of the stages with a number after it. So we have prophase 1 and then we have prophase 2, the second time it happens. And what happens in prophase 1 is the homologous chromosomes find each other. They align. So we call that synapsis. That's when the homologous chromosomes find each other and they align together. And that's what's happening here. Okay. So the two that are equivalent, that are homologous, like the mommy chromosome number 1 and the daddy chromosome number 1, they end up finding each other. How they do that, I don't actually know. I'm sure some smart scientists know, but how they do that, but they do. They manage to do it. And then what happens is there's actually some proteins that form around them to kind of bind them together to make sure they don't come apart. So when they get held together, that's called the synaptonemal complex. The synaptonemal complex. I cannot believe PowerPoint spelled that correctly. The synaptonemal complex. It like knows what that, I'm so tickled with PowerPoint that it knows what I'm saying and spells it correctly. That's awesome. You can see it on the bottom there, synaptonemal complex. That's basically just the two homologous chromosomes stuck together during synapsis. Okay, so that when they get moved around in prophase and you're going into metaphase, that they don't get, you know, they don't get separated, they don't get yanked apart. That's the point. And then in the first division, what happens is the X's actually stay intact. So the entire sister chromatid complex for the maternal one and the entire sister chromatid complex for the paternal, did I say maternal? Maternal and paternal one or whatever, they stay together. All right, so these guys here are still together, and these guys here from mom are still together. And the reason why they're still together is because at this point, there's little tiny proteins that hold them together that are called cohesins. We learned about cohesins during mitosis. Those same cohesins are right here, but they didn't get split up, and they're not going to get split up during anaphase. So during anaphase, the cohesins stay on there and hold those sister chromatids together so that the entire X gets moved to the other side, and the entire X from over here gets moved to that side because the cohesins stayed there. Does the cohesin thing make sense to you? Are you with me? Like think about little like wires or little strings that are tying them together, like shrink wrap. Think of like shrink wrap tying those things together, and they didn't get cut. They stayed intact, and then it allowed the X to go one way and the other X to go the other way. I thought I explained that really well, but I'm not sure based on your faces. I hope I did. Also, if you could turn your cameras on, maybe just for me, so I could, that would make me more efficient, because then I can kind of see if you guys doing okay without having to, you know, always get you to say something or whatever. Although I would love it if you guys said stuff. I would love to hear from you. Would make me very happy. But I mean, if you can't, then you can't. And at least I can see your face. Thank you. Almost everybody has it on. I appreciate that. Thank you. Okay, so there's a really smart scientist called Watanabe. He's Japanese. And Watanabe discovered what protects the cohesins. He found this molecule in the cells that actually protects and guards the cohesins and prevents them from getting cut by those enzymes that split them. And he was so impressed with this molecule that's protective, like a guardian, he called it a shugoshin. So the shugoshins, that's Japanese for guardian spirit, the shugoshin protects the cohesins from getting cut. They're like guards that stand around the cohesins and, you know, they fight off those evil enzymes that try to cut the cohesins, and they protect them during meiosis 1, so the cohesins can keep those sister chromatids together and they can get moved as an X. So that's kind of a cool story. I kind of like that. I think I don't remember all the details, but I read about Watanabe, and I think his wife was a scientist too. I think that's the one I'm thinking about, and they both were like real brilliant biologists. But that was a long time ago that I read about them. I don't remember all the details. I just like I like the names. I kind of like the name Shugoshin. I think it's kind of awesome, and the fact that it means guardian spirit, I think, is really cool. It's like it's kind of like an adventure story. Miss Certain, is everything okay there? Are you okay? Are you getting attacked?
Yeah, my cat.
Oh, that was your cat. Okay, all right. All right, so the second division then, in the second division, the X's get split. So now we're going to split. Now the Shugoshins gave up their job because eventually the sister chromatids do have to get separated, and then we make those haploid cells. All right, we're going to take a look at that in a little bit more detail. So what we got happening here in the beginning is the homologous chromosomes find each other, and we have synapses happening. The other thing that really you ought to know about is that during synapses, here's what happens. You get crossing over. So crossing over happens where the two sister, or these sister chromosomes that are closest to each other they're kind of goofing around a little bit, and then they get stuck. You know, your mom always told you, like, don't do that, your face will get stuck like that, or don't do this, it's going to get stuck. Well, these guys actually did get stuck. So the one's like hand got stuck to the other one's arm, and the other one's hand got stuck to the first one's arm, and then they got separated like that. So now all of a sudden we have a maternal chromosome here that actually has a little piece of the paternal part on it. So anyway, when this happens, we call that chiasmata. They form. So the stuck together homologous chromosomes are called chiasmata when they're stuck together. So that's confusing because it's like, oh, the process is synapsis. The thing that forms when they cross over is chiasmata. Anyway, lots of weird terms that need to be kept track of here. The crossing over thing, or the chiasmata thing where they combine, actually helps them stick together too. So the synaptonemal complex helps, and the fact that they're stuck together like that during crossing over helps them stick together. It keeps them together all the way into metaphase, which is kind of awesome. And then in metaphase, they split, and that little blue part here gets stuck on one side, and I'll show you that in a little bit more detail. All right, so here the Shugoshins are protecting. The Shugoshins are now protecting the cohesins. And then notice when they get split right here, that whole X gets pulled over, and here the whole other X gets pulled over because the Shugoshins protected the cohesins. So that's kind of awesome. And that's necessary for meiosis because you want to get that DNA divvied up correctly. All right. So notice we have 6 over here. We only have 3 now in each cell over here. All right, then we divide again. So the second metaphase that we get looks just like mitosis, except now instead of having 6, we only have 3 lining up. So you know it's not mitosis. It looks exactly like mitosis, except half the number of chromosomes. So if this was human, in this we'd have 23 now instead of 46. For mitosis, we'd have 46 lined up in the middle, all the way down. And that's not what we have here. I gotta be careful with what I draw that it's not so easy to erase because I'm in present mode. All right. I don't need to go into like centrioles and centrosomes and spindle fibers and all that. That's the same as mitosis. So review that if you need, if you're kind of lost on that. But then they get finally, so now the Shugoshins gave up their job. The Khoisins are all cleaved. Now the X's get separated. So this one goes this way. This one goes the other way. And we got this happening here. Okay, so now we only have one of each chromosome, only one chromatid left that goes to each cell. And we only have 3 and 3 now. You might say, well, wait a minute, it was 3 and 3 last time. But yeah, but it was 3 times 2 chromatids. It was 3 X's. Now it's just 3 single sausages, no more X's. So we're actually reducing at this point. Okay, so after that happens, we say these guys are, we say they are haploid. Okay, or N. All right, by the way, 2N means diploid and N means haploid. N is just the number of chromosomes. Okay, if we were going to draw this ourselves, we could do this. Okay, so I'm drawing my synapsis spiel. I'm drawing my chiasmata, whatever, forming here. And I'll just do it with 6. So this would be prophase 1. This would be metaphase 1. This would be anaphase 1. Now the X's are getting intact, getting pulled to the opposite ends. Okay, cohesins are still intact. Okay, we're getting ready to split now. So what we're going to do is, here we're going to split telophase 1. What happens is it doesn't go back into interphase at all. Now we only have 3 X's in each nucleus. Okay, so at this point we're haploid. So after this point on, we're now haploid. Once this splits right here, cytokinesis. All right, so then we're gonna go back into, so we could call this, okay, let's split them actually. So now they split. This is kind of redundant because this goes right into prophase 1 now, and we have no interphase in between. So we do not duplicate the DNA again because that would defeat the purpose. I'm sorry, not interphase, prophase 2. Gonna go a little smaller now. It's kind of nice to draw this out. You might say, well, that's a waste of time to draw this out, but I think it really helps like solidify the concept when you have to think about like, wait a minute, do I draw 6 here? Do I draw 3 here? Are these X's? Are these not X's? The nucleus is gone here, obviously. This is metaphase 2. So I think it might be nice to draw it out just to, it's not like I'm going to make you draw it for a test or anything like that. It's not like you got to memorize it for the College Board, but to get the concepts, it does help. We got anaphase. So now finally the sister chromatids are split up. Those cohesins are gone. All right, and then finally we get, kind of ran out of space here. Notice the number of chromatids now, or chromosomes in each nucleus. We only have 3 left. We started with 6. We only have 3 now. So then this splits again. So this would give us 2 cells, obviously, with 3 each. All right, so that's how we reduce the chromosome number. Everybody clear on that? Everybody good to go? Good to go. Good to go. We got a thumbs up. Thumbs up. All right. The thing I'd like to talk about now, well, actually, hold on 1 second. Let's not look at this yet, because this right here, this stuff with independent assortment and everything, we're going to take a look at in chapter 14. So I'm going to talk about independent assortment and the whole crossing over business and all of that stuff later. Let's worry about something else right now. So mitosis versus meiosis, that's something you probably want to look at, but be nice to know the difference. You could make a Venn diagram if you wanted to. Like, what do they have in common? What's the same? What's different? Obviously, you know, 2 divisions. What's the chromosome number of the daughter cells? In mitosis, it produces 2 N diploid daughter cells that are clones of the mother cell. In meiosis, it produces haploid N daughter cells that are different. They only have half the DNA, all of that stuff. Homologous chromosomes never find each other in mitosis. They only do that in meiosis. So synapsis never happens in mitosis. It only happens in meiosis. All that business. All right, so check that out. That's nice. I want to talk about this a little bit here. I want to talk about the combinations of gametes we can get depending on the chromosome number. Okay, so this requires you to do a little bit of math. So if you have a calculator or a phone that has a calculator on it with an exponent button, like an not exponential, but the up caret button, you know, exponent, y to the x kind of deal. We're going to do a little bit of math. So let's try something. If I have, that we're not going to worry about right now. Let me insert a new slide. All right. So this will give you a little time to get your calculator ready. So let's do this. Let's say I have a cell or an organism that only has 1 pair of chromosomes. So, and it's sexually reproducing. This is kind of fake because I don't even know if there is one that only has 1, but let's just say hypothetically there's one that has 1. So you got your cell and you got your mom chromosome and you got your dad chromosome. Now I'll just, instead of just constantly switching colors, which is a pain, I'll just have the dad one filled in with ink and then the mom one not filled in, so it's easy to keep track. So let's think about this now. How many different kinds of gametes can this organism make? Let's assume no crossing over. We're going to assume no crossing over. We know there's crossing over that happens all the time. Crossing over is extremely common. It happens basically every time, but we're going to assume it doesn't happen just for fun. How many different gametes can this one make? So we divide, we end up with this and with this. So how many different kinds? I'm not looking at total number. Obviously we'd get 4, okay? Because this would be like a, you know, sister chromatids, they divide twice. Let's not worry about that right now. So how many is that? We got 1 maternal one, we got 1 paternal one. How many different sperm or how many different eggs can this organism make?
1, 2.
Well, they can make one that looks like the white inside, and then they can make one that has the black inside. Does that make sense? So it's 2 different kinds. Does that make sense? That means half of the sperm in that organism will be this kind. The other half will be this kind. There's only 2. Still with me? All right. So now let's try it again, but now let's do it with 2. So let's say they had 2. So we got this. Well, if you want, I can draw them all in. I was just being lazy over here, but I'll draw them all. So here's chromosome number 1. Here's chromosome number 2. So they have 2 sets of chromosomes. Okay. The thing is, because of independent assortment, which I said I wasn't going to talk about today, but I'm still going to talk about it, they could line up a different way. let's take a look at the other way they could line up. And I think you guys might already know this from freshman year, or just from common sense. Even if you've never learned this, just like common sense might tell you, is there another way these guys could line up? Let's just do the first one the same. Could number 2 do something differently if number 2 does not care? I read your thing, Claire. I'm not sure I fully understand it, but it sounded really epic. Tell me the other way these guys could line up if chromosome number 2 literally does not care about chromosome number 1 and doesn't care what the first one did. It just does its own thing. What else could chromosome number 2 have done in terms of the black ones and the white ones, the maternal and the paternal? The whole idea of independent assortment is that the second one doesn't care about the first one. Either I didn't explain that very well, or you guys just don't feel like answering.
The black one could come first.
Can you say it louder?
The black one could come in front of the white one, so it'd be like backwards.
So could it be like this? Could this happen? Yeah. So that's another way they could line up. And that's what independent assortment is. Mendel taught us about that, is that these two don't care about these two. Okay. So if we divide here and we divide again, we're going to end up with some gametes that have all maternal. We're going to end up with some gametes that have all paternal. We're going to end up with some gametes that have one maternal and one paternal. And we're going to end up with some gametes that have one maternal and one paternal, like this. So we have 1, 2, 3, 4 different possibilities for different combinations of gametes. So the formula that some real smart scientist came up with is the way we figure out the number of combinations is 2 to the n. And the n is the number of chromosomes. So if we only have 1, that would be 2 to the first is 2. We said that before, that if you only have 1 chromosome, you only have 2 possibilities. Okay. I'm going to write Bill Gates and tell him to put an undo feature into PowerPoint so that I can easily undo this instead of having to go to eraser each time. Okay, if I have 2, it would be 2 to the second. What's that? 2 to the second is?
4.
4. If I have 3, shout it.
8.
8, et cetera, et cetera, et cetera. So that's what happens. So another way to easily show that, like if you're wondering like, oh, what about the 3? You know, what are you talking about? How does that work? Well, let me show you. I will show you. Other way to show that would be just like this. Actually, yeah, let's just do it like this. Oops. Are you with me here? Do you see what I'm doing? And I'm not implying like dominant and recessive here. I'm just saying big A, little a is maternal and paternal. That's all. That's all I'm looking at here. We're not going to worry about dominant, recessive, and all that stuff yet. That's chapter 14. We'll worry about that then. I'm just showing you like how many different combinations I can do. This is just a lot easier to draw than drawing these things here, you know, because those take me a while to draw. So let's count them. We have 3 chromosomes. I'm sorry, yeah, we have three chromosomes. We have A, we have B, we have C. So we're looking these letters as the chromosomes, not as genes necessarily. All right, and we have these many different combinations. 1, 2, 3, 4, 5, 6, 7, 8, which is what we got. Now humans have 23 sets of chromosomes. So now it's 2 to the 23. Now it's time for your calculator. How many combinations of gametes can this human make? Assuming no crossing over, which we know happens, which would make it even bigger, but let's just type that in the calculator. So the way you would type that in is 2, up caret, and then 23, and then enter. So let's see what you get. Amra says, and Vance says 8,388,608. Let me check my notes, see if that's right. I'm sure it's right. It sounds right. I have it here somewhere in my notes. I was lazy and I called it 8.4 times 10 to the 6, which is right. So it's 8 million something. Okay. That's how many different sperm you can make with 23 chromosomes without crossing over, without any of that stuff happening, which means if crossing over happens, you can make a lot of different, way more combinations. But sperm aren't people and eggs aren't people. Okay, so to make a people, to make a Volk, by the way, you know, Volk means people. I can actually say that, to make a Volk. The German translation of Volk, or translated into English, is people, literally. So see, I am a people person after all. You doubted it. You doubted it. I love the faces as I banter like this, and I love the faces of just complete, utter despair. You know, here I'm making hilarious jokes. I'm telling you little anecdotes. I'm making this super fun, and all I'm getting is like, so thanks for that. Thanks. Merry Christmas. Anyway, we're not sperm, we're not eggs, we're people, and a people has sperm and egg combined, which means now in order to figure out the odds of two siblings being identical, not just two sperm or two eggs being identical, but the odds of two siblings from the same two parents without counting crossing over, no crossing over even considered, is 2 to the 23 times 2 to the 23. So it's that 8 million times, okay, squared. So whatever that is, go ahead and do that. 7.0 times 10 to the 13th. So 7.0 times 10 to the 13th is going to be, I got to think now, a million is 10 to the 6th, a billion is 10 to the 9th, a trillion is 10 to the 12th. This is 71 in 70 trillion. Are the odds that you will have a doppelganger doppelganger is maybe a word you've heard, like a clone, within your own family I'm talking about, not like out in the world. I'm talking about a sibling from the same parents. Let's say you're born and you're you, and now what are the odds your parents are going to have another kid that's exactly the same genetically as you, not even counting crossing over or anything like that? The odds of that happening is 1 in 70 trillion that your parents would produce another child that was genetically identical to you. That's pretty slim odds. So we see that siblings are somewhat similar a lot of times. They kind of look the same, but they also have a lot of differences. And the reason they have those differences is because of this shuffling, this independent assortment business, this shuffling of the DNA between the maternal and paternal chromosomes, that we get this nice, beautiful mixture with each child, with each offspring. And that's true for any sexually reproducing organism, but we care about humans the most, so we talk about humans. So all that makes sense so far? Everybody with me? If we throw crossing over into the mix, the numbers become staggering. It becomes unmanageable because crossing over increases the variety so much that these numbers are not even in the ballpark. The odds are becoming way, way, way less even. So yes, we have a very high number, but the odds are very, very small because this is a fraction, right? It's 1 over 10 to the, what did I say it was, 13th? Okay, or 7 times 10 to the 13th. It's 1 over that. So the higher that number gets on the denominator, the smaller the odds get that would ever happen. So now people, my sister and I look like the same person at different stages of life. Yeah, you might have quite a few similarities, but you're also going to be, you know, you're different from your sister. You know there's differences. Physically, personality, all of that stuff. Anyway, people talk about like doppelgangers, like you see somebody out in society that's not related to you, that's your clone, that's the same. Like Princess, what's the, you know all the Netflix stupid Christmas movies they have, like with the princess this and that, the Princess Switch. Have you seen the Princess Switch? We watch all those at home. Like every year we watch the new Netflix, like crazy, stupid, the Christmas Prince and all that. But in the Princess Switch, you should watch it. It's amazing. And when I say amazing, like it's so bad. It is so bad, it's good. That's what I'm talking about. This girl like meets somebody else that looks exactly and sounds exactly like her, and then they switch roles or whatever, and it, you know, pandemonium ensues and whatever. But anyway, and of course, then you know they all fall in love with the other, you know, the other boy that they ended up. Oops, sorry. They fall in love, of course, you know, and then happy ending. Oh, and I just spoiled it. Now you don't have to watch it. Okay. Anyway, square things. Did you just call my beloved Punnett squares square things? That's what you called them? That is blasphemy. How dare you? Who are you? I already put worksheets on Google Classroom that you can do over break. Yay! You can do Punnett squares. All the way from freshman level, easy peasy, and all the way to like make you want to throw up in your mouth level. We got all sorts. Pungent, pungent squares. Killing me. All right, that kind of sums up chapter 13. I did a pretty good job getting through that. Hey, that was all right, wasn't it? Okay, any questions? Please do ask me questions, because I know I go fast and I know I'm hard to understand sometimes. You are literally the Grinch ruining Christmas for us. I literally scared you never answer me. I literally scared you never answer me. Okay. Oh, okay, so I just got to go back and find it. I'm sorry. Wait, personality traits are not passed in genes though. Yeah, sure they are. Personality can be in genes too. That's multifactorial, which means it's environmental and it's genetic. It's both. There's a bunch of traits that actually are controlled by genes and by the environment both. A good example of that is like, have you ever seen like peonies, the flowers, the big round blooms that they have? If you put them in acidic soil, they'll be like reddish, pinkish. And if you put them in basic soil with a high pH, they'll be blue. So the color is determined by genes and by the environment. And personality is like that. So if somebody is like raised a certain way, that can mold their personality a certain way. And you know how it is, like if kids are abused or whatever or neglected, then they become, their brains are damaged by that. I mean, not physically necessarily, but their emotional development is damaged and they can have a lot of psychological issues. So that is the environmental part. It's not necessarily genetic. But yeah, part of the, like my father and I are very similar. I'm starting to become painfully aware where my wife sometimes has to say to me, okay, Heinz, because my dad's name is Heinz. And they like, and when she says that, it hits me like, oh my God, I'm just like him. Like, wow. Anyway. Heinz is in top form this season. He's, let me tell you, he's, yeah, it's, yeah. Anywho, anything else? Okay, let's take a look at Google Classroom, please. We'll check that out and look at all the wonderful things I posted for you. I was so generous. I am a giver. I'm not the Grinch at all. These are all wonderful holiday presents for you. There's chapter 13 stuff that I had up there from a while ago, from the 18th. Now there's stuff that's up here. There's chapter 14 stuff. So that's genetics, Mendelian genetics, you know, with the Punnett squares and homozygous, heterozygous, dominant, recessive, all that business. So there's easy freshman, and there's a little bit more advanced stuff. These are actually the easier ones. And then these are a little bit more advanced. You probably won't know how to do all these because there's stuff in here we haven't learned yet. And then here's some chapter 15 problems with a lot of things we haven't learned yet. So we'll get into that in the new year. And if things go well, we're going to get all this genetics business done, and then have a wonderful final in the end of January. So let me draw attention to something else for you, and that is this: the midterm free response question review. This is quite the beast, and yes, this will cause you anxiety. It's causing me anxiety, and I'm the one who wrote it. So this is lots and lots of fun questions. A lot of this stuff is stuff you've already done, like redundant questions that you've already answered, because they're like review questions that I gave you for some of these tests that we had. Okay, so, oh, describe the stages of mitosis, you know, well, okay, whatever. Well, here's some genetic stuff that I haven't covered yet. So the genetics of human blood type, like the IA, IB, and all that stuff. We'll cover that again. I'll go over that with you. A lot of genetics things, so we haven't really covered that yet. So if the genetic stuff is throwing you for a loop right now, don't worry about that too much. But okay, one thing I did not teach you is standard error the mean. So we might still do that in the new year. I might go over that a little bit with you. I mentioned it briefly for this last test, and I put a question on there where you didn't have to do calculations. You just had to interpret. But I might show you the calculations because they're kind of fun. And then, oh, well, this looks familiar. I know this. You know, I know how to do all, you know, so there's things that are like signal transduction, there's cell stuff, there's cytoskeleton stuff, enzyme activity, protein structure, respiration, photosynthesis. So I would say that I'm going to pick some of these for the midterm. And if you do these and you at least go through them, you should be in good shape, because I'm not going to throw you any curveballs. I'm not going to give you any surprises on the midterm. I'm going to give you something from here. Do you have to do all these and turn them in? No, but it might be a good idea to spend quite a bit of time over break on these. That would be nice. So you don't save all your studying until, like, you know, a couple of days before we have the midterm. When is our midterm? I don't actually know. I have to look at when our, I don't even know if they're going to have a proper exam week and how they're going to do that, but it's somewhere like in the 3rd week of January or something like that-ish. So that's available for you. Now we only have 20 minutes. I want to play Kahoot. Is that cool, or do we need to talk about anything else? Is there anything I'm forgetting that I'm supposed to talk to you guys about? Because sometimes I forget stuff. Tell me if I'm forgetting something. My sister and I don't look related. Yeah, I don't know. Sometimes that happens. Well, in my family, you know, I'm way more handsome than my brothers, so that's always confusing. Like, are they really related? I don't know. Okay, let's do it. Go to kahoot.it. Kahoot.it. That's what you're doing. And go. I'll tell you the code in just a second. All right, for this, I'd like you to have the cameras on, and I'd like you to like give me nonverbal and verbal feedback during the thing if you appreciate this. So I want to hear like noises. I want to hear like chuckling. I want to hear like noises of like that you're deep in thought, stuff like that. Everybody clear? Everybody good to go? All right. All right, let's play it. You guys know how this works? The faster you answer, the more points you get. Okay. And you have to click on the colors on your, if you do this on your phone, you can click on the colors on the screen. That might be easier than on a Chromebook, but you have to look at the questions on the screen. It's kind of dumb. Like, it doesn't give you the questions on your screen, only on mine. So kahoot.it, and then enter 212-2819. So it's 212-2819. Pigment squares. You guys are weird. Oh, I hear music. You hear music on your end? I hear that music. IG, like he goes from bad to good. Wait, I can see it. The good Grinch, that's Volk. There was a good Grinch? Still evil, but good. Kahoot time. Do we need this to do this over break, like the review? I would work on it over break, but it's not. I'm not going to collect it. 2021 exists. Yeah, soon it's going to be 2021, and all our problems are going to be gone because 2020 will be over, and everything will be like super awesome. So I'm totally looking forward to coming back after break. It's going to be amazing. A free trial. Now we're going to sign up for the paid version. Okay, way too much chat. I can't read all this. Oh, the Grinch. Is everybody in? I got 25. Don't I have more people in here than 25? Oh no, we got everybody. Everybody's in. All right, cool. We're gonna start. Here we go. Kahoot. Disulfide bridges ionic bonds and hydrogen bonds between our groups. Ah, nice. Somebody thought it was secondary. 9 of you thought it was secondary. Did you forget all that stuff from the beginning of the year? That's still going to be on the final. You better study it. Angie is in the lead with 950. We got Elena, Leilani, Vincent, and Maxwell in the top 5. Nice. Competitive inhibition is when the inhibitor binds at 3 seconds. Put your answers in. Noise. Most people got it right. Excellent. Active site. Beautiful. Whoa, we got massive shakeup here in the top 5. Holy cow, that was brutal. Leilani in the lead. We'll see how long it lasts. Vincent is in second. My goodness. Muscle contraction and cytokinesis. Okay, quite a few people thought it was microtubules. Study that cytoskeleton. How are we doing? Whoa, she kept the lead. Paige is in the game. She is moving on up. Hey, if you don't want my commentary, tell me. Otherwise, I'm just going to keep doing stupid commentary the whole time. Anyway, next. An animal cell suspended in a solution. Net water movement will be... So the cell has -3 bars potential, and the solution has -12 water potential. Two seconds. Oh, most people got it right. And nobody put the bottom 2, so thank you. That means you guys are actually paying attention. A little bit of a shakeup, but looks like Abigail is moving up. There's like a triangle next to her. I don't know what that means. I think she's gaining. Honey does not spoil. $19 people got it right. Nobody put the green one. Come on. You guys get that joke or no? You don't get that joke? Honey is this app you can get on your phone or on your computer that like gives you coupons. It's super intrusive. Everything you do, and all of a sudden like Honey pops onto your screen. Like, the crap. I stupidly installed it. Now it keeps, I don't know how to get rid of it now. Leilani's still in the lead. Abigail in second. Vincent, Paige, and Angie on the move, looking for that first place spot. We shall see how it goes. Leilani's probably getting nervous. Protein kinase C is activated by, easy. These are like middle school questions here. 3 seconds. Ooh. What happened? What? Can I tell who put what so I can implement some consequences? 20 people put the wrong answer? Oh, is it because it's like C, cyclic AMP starts with C? You figure like, oh my goodness. You forgot all of chapter 11? Okay, this hurts me. Didn't answer. I'd almost rather not get your answer than this. This cut me deep, people. Ouch. Julia moving up into the top 5. G protein coupled receptors will activate their G protein by, do not get this wrong. I gave you extra time on this one. I gave 30 seconds. Nice job. Most people got this one. Appreciate it. Although 5 people did think it was a phosphate. 0 people thought it was RTK. Nice. Oh, Leilani, no. Dethroned. The higher they fly, the deeper they fall. Abigail in the lead. Page slash. Is that supposed to be a threat page? Page slash. Are you trying to hurt me? Where is she? Oh, yeah, clearly a threat. Julia, Leilani, and Angie. I got a question. Where are the boys? Where are the boys? Do we even have any boys in this class?
I'm on my way, Mr. Volk. I'm almost there.
Okay. I'm counting on it. Got to represent testosterone. Amplification is possible when? This is a tough one. I gave you 30 seconds for this one. Okay, most people, I shouldn't say most people, but the highest number was the right answer. So nice job. But let's see if this shuffles things a little bit. There's quite a bit of shuffling, but Abigail remains in the lead. Leilani is back. She is back for blood. Vincent, Angie, Paige. Vincent, thank you. Thank you, Vincent. Thank you for representing my species. All hope is not lost. Moving on. Self-suicide during development or after damage to internal components. We did have one that thought it was atherosclerosis. Interesting. I'm not going to say who it was. I don't know who it was. Do not identify yourself. Do not. By the way, did you appreciate this part? This is basically me at home. Series of signals pointing to his obsolescence. Anyway. Okay, you could laugh out of politeness, out of mercy, for me, for my feelings, you could laugh. Just saying. But apparently my self-esteem doesn't matter. Apparently my feelings don't matter. But that's okay. I get paid to be here. I don't need to have feelings. I understand. Good job getting that one right, though. Abigail in the lead, Leilani, Vincent, Angie, Paige slash. Schmooze form in what organism when love is in the air? Yeast. Very nice. Very nice. I don't understand you mislicked. Not supposed to lick the thing. I don't understand the comments. Confess who you are. I don't get that. And I don't understand OFC, so I'm sorry. Is that supposed to be like an insult or something? You are the problem for us? Okay, thank you for that. Is that a sea lion? No, that was a shmoo. Can I go back? Oh, I never told you about shmoos because I made you learn those on your own. Those were like a cartoon from the 50s, like a comic strip in the paper. I accidentally clicked blue. Yeah, that happens. All right, moving on. Most plentiful protein in the world, also great at fixing carbon dioxide in plants. By the way, the answer is in the picture. All right, most people got it right. Some people thought it was Ruby P, but Ruby P kind of sounds like Rubisco, so it's real close. You know, that's like the second best answer. Nobody put Ruth from Ozark. I don't know if you guys watch that show. She's crazy. Couldn't see the picture. Sorry. Abigail, Ilani, Vincent, Julia, Maxwell. We got a 3-to-2 ratio here. Let's move on. Thylakoid membrane is similar in function to. The thylakoid membrane is most similar to which of these? Hold on, I got to do something real quick here. All right. Oh, it looks like most people got that right. Nice job. Way to go. And we have no shifting at all. Everybody stayed the same. People on below the top 5, you've got to catch up now. You got to get some of these right, and you can boot some of these people out and gain that glory. What will happen to light reactions when you remove CO2 from the environment? I believe in you, Ensign. You can do it. See if you can refresh, Noah. See if that helps. You should show us your dancey dance. I don't know what that means. I don't know what a dancey dance is, and I still don't know what OFC is. 22 people got it right. Nice job. Way to go. And oh, we have shifting. We have shifting. We got Cadenze in the game here. We got Abigail, Ilani, Vincent, Maxwell, and Kadai. Pukey a Clown is at South Carolina State Fair making. By the way, if you get this wrong, instant dismissal from class. Somebody put cream puffs. Somebody put cream puffs. Who was it? Who was it? How dare you? No shifting. Everybody stayed in the top 5 the same. Moving on. Big A, little a by Big A, little a. What are the odds of getting homozygous recessive offspring? Little a, little a. What are the odds of little a offspring? This is genetics, what's coming up. Looks like most people got this one. Thank you. And we got a little bit of shifting. Predict the phenotype ratio of that dihybrid. Double hets, both sides. Pungent squares. They're very smelly. 5 seconds. Get those answers in 1 second. Let's go. Most people got it right. Some people thought it was 93-4. We're going to learn about it 93-4 in this class. It's very interesting and very odd. Oh, we have shifting. Vincent is in second place. Leilani, Katie, and Maxwell trailing behind Abigail and Vincent. Very nice. We have 3 questions left and 3 minutes. This is a tough one. I think I gave you 30 seconds for this one. What are the odds of inheriting little b from the father? Well, maybe it's not that tough, but it's sex linkage, which is always, you know, a little more confusing. The odds of the son inheriting little b from the father. Some people thought it was half, and then 11 people got the right. Yeah, 0. The answer is 0. It's impossible for him to inherit that from the father because the father only gives the son the Y chromosome. There's nothing on it. We'll go over that stuff. Don't worry about it. You're not like expected to know that already. Although we did talk about it freshman year. I think all the bio classes talked about sex linkage. I hope they did. 17. So we have one more. Oh, last question. This is the most important question. Double points. Who is not your favorite AP Bio teacher? If you get this wrong, there'll be consequences. Keep in mind, it says who is not your favorite AP Bio teacher. Sophie, that's that picture I emailed you. Remember? People got this wrong. Okay, I will find out who put these wrong
Mr.
answers.
Volk, I made it to 5th.
Oh, nice place. Nice. I'll take a look at that in a second. No, I'm confused about why people think Martin Volk is not your favorite AP Bio teacher. I'd like to know who your favorite AP Bio teacher is. And then somebody else thinks that Volk is not their favorite bio teacher. I think some of these people are confused. Confused. All right, here we go. Let's see what the results are. Cadenze in 3rd place. Leilani, 2nd place. Abigail in 1st place. Nice job. Way to go. And a couple of boys in 4th and 5th. They know their place. They know their place. All righty. It looks like class is up. If anybody is staying for TSS, that'll start at 1:10. I have some issues I need to talk to some of you guys about, or maybe to all of you guys about, but I'm not sure now is the time because class is almost over and I don't want to keep you late. But if anybody wants to help me out, stay on for a couple of minutes and you could do me some favors, but you don't have to. You can leave and have a good New Year's. Merry Christmas, happy Ramadan, happy Kwanzaa, happy Hanukkah, all of that stuff, and happy New Year. Have fun with all that work over break. Email me if you need help or leave me a note on Google Classroom. See you later.